Hello all, welcome to this week's session on evolutionary tree reconstruction. So, in this session of algorithmic thinking in bioinformatics, we are going to be looking at a set of samples and then sequences, genome sequences from these samples or protein sequences from these samples, and then we would like to know how to construct evolutionary trees to understand how the sequences might have evolved from a common ancestor. This is a very basic problem in evolutionary biology. We would like to know the tree of life which relates all the species, or in terms of clinical use, in terms of the pandemic that we just got out of or is still going through in different countries in different phases, this COVID pandemic. This will give you a way to understand the origin and the evolution of viruses or bacteria that cause infection, right. So, this is a very a basic evolutionary biology question as well as a very pertinent question for clinical practice and in terms of pandemic control as well. So, we will be looking at again how to formulate this problem as a computational problem using the concept of trees to relate a set of sequences, and how do you actually reconstruct these trees from a set of sequences and the distances between those sequences. And so, those are called as distance based phylogeny algorithms and neighbor joining algorithms. Okay. We look at distance based phylogeny as the problem, and then there are different algorithms for the problem, and neighbor joining is one form of algorithms to construct an evolutionary tree out of a set of sequences. Okay. In order to put it in the context in terms of other topics seen in the course so far, we have addressed different biological or medical related questions in this course and their algorithmic solutions, right. And we can think about it as largely in two categories. If you wear your computer scientist hat, we can group the computational problems or algorithms into broadly two categories. One is the set of reconstruction algorithms, right, where you are given some measurements from some sequencing technology or other experimental measurements, and then these measurements are giving you different bits and pieces of an underlying object, and we want to reconstruct that object. Okay. This is what we saw, for instance, in genome assembly algorithms. Okay. And in this particular topic on evolutionary trees also, there is an underlying evolutionary tree which relates all the sequences of interest, and we are given just the sequences, so some kind of measurements on the sequences and the distances between these all pairs of sequences, and we want to reconstruct the underlying evolutionary tree, right. So, those are the reconstruction algorithms, and the other algorithms are more familiar to us as computer scientists. We are given a data structure, we are given the input, so we are given a graph and we want to find the shortest paths in the graph. We are given a string and we want to find the most frequent k-mers in the string, or we are given two strings and we want to find the best matching or alignment between those two strings, right. So, those are all what I call it as searching or matching or other analysis type of problems which we as computer scientists are most familiar with, and bioinformatics has a lot of interesting applications for the reconstruction problems, and reconstruction algorithms have been developed also, and so that I hope provides you this a well rounded perspective of what data science could do, right. So, data science is not about analysis of existing data structures or existing data in terms of string matching or kind of shortest paths in graphs or other kind of problems, but you can also look at measurements from these technologies and try to build the underlying data structure that could have generated these measurements. Okay. So, those are the reconstruction algorithms. And like I said, in this particular session we are going to show a reconstruction algorithm of reconstructing the tree relating a set of viral samples, and thereby infer its origin and how it has transmitted from one person to another or one region to another region. And so, this would be of much use. It is pertinent because of the recent pandemic. So, this would be much use in terms of understanding the genomic aspects of how a virus spreads during a pandemic, and it will also give you some idea about what are the causative agents for a disease that is spreading rapidly, and some ideas on how we may be able to control the disease. And to address this problem, we are going to look at an associated computational problem, right. Given a set of sequences, how do you construct this tree, how do you formulate it concretely, and then some algorithms towards solving the same. Okay. Let us look at these 2 points a little bit more carefully, so that we put it square and front in the context of this particular course, right. Like I said, as computer scientists we are mostly looking at analysis problems, and but we would also, as bioinformaticians, we also look at this different aspect of data analysis. So the role of data science or data analytics in genomics and bioinformatics is this contribution of these different types of problems and algorithms for reconstruction, or it is also called realization. You want to come up with a data structure that realizes the data that you have measured. Okay. In terms of genome assembly, the data that you have measured is all the k-mers in a large string or a genome, right, and then using all the k-mers you want to construct a single genome that will contain all these k-mers, right. So, you are trying to realize a genome or reconstruct a genome whose k-mer composition is matching or exactly the same as the k-mer composition that the sequencing instruments have given you as your outputs, right. Similarly, there is another problem called reconstructing a peptide from protein experimental data. So, mass spectrometry is a way of measuring what are the proteins present in a sample, but again it does not give you the full protein sequence, it gives you some other form of measurements of proteins. We will look at it more detail in a later week in the course. But given those measurements from mass spectrometry instruments, we want to reconstruct what peptide could have given rise to those measurements. Okay. The same way that the genome assembly, we want to reconstruct a genome that could have given rise to these reads that we have, right, or the k-mers that we have. In this peptide reconstruction problem, we want to reconstruct a peptide that could match a set of measurements from the protein mass spec instrument. Phylogeny, or this evolutionary tree, also known as phylogenetic tree or phylogeny, right, which is the topic of this week, is also something similar. We are given some distances between all pairs of species of interest or all pairs of viral samples of interest, and then we want to find out a tree that relates the evolution of these set of sequences. Okay. So, the other analysis problems, as computer scientists, like I said, we are more familiar with these. We do not have to worry about trying to reconstruct the object that we want to study. The object is given to us, right, and then we are asked some properties about the object. So, for instance, you are given a DNA segment and then you want to find the origin of replication in that DNA segment, and there we saw that we could address this using this frequent k-mers in a word problem, where the word is a string that is representing your DNA, and then you are looking for k-mers that occur frequently in this particular string, right. And that is like in some form a searching problem, searching the most frequent k-mers. Sequence alignment is also a problem where two sequences are given to us, you do not have to reconstruct those sequences, they are just given to us, and then we want to know what is the best alignment between those two sequences in terms of edit distances or longest common subsequence or some form of more general scoring between those two sequences. That is a way to understand how evolutionarily related these sequences are, right. So, sequence alignment will play a role in constructing the distance matrices that is going to be the input to our evolutionary tree reconstruction algorithms. Okay. And we have also seen suffix trees and suffix arrays as data structures for indexing a large genome, so that we can match a set of numerous set of short patterns, right, short reads to the large genome, right. So, that is also a case where your reference genome is given to you, you do not have to reconstruct it, the reads are all given to you, and then you just need to match one against the other. Okay. And we will also see later on about this motif or pattern finding in a set of sequences, right. You are given a set of sequences and you are asked to find some pattern that frequently occurs across these set of sequences. All of these are problems in which you are given the object that you want to analyze, and you want to find a property of the object or a subset of the object, or we want to do some searching or matching or other types of analysis problems. Okay. So, these are the two broad categories of problems, computational problems that arise in bioinformatics and genomics, and that we have been looking at. Some of them we have already looked at, and some of them we will be looking at in the future weeks of this particular course. And then the techniques that we see here also, as computer scientists we may have seen many algorithms on strings, trees, graphs and so on, but still there are certain algorithms in genome assembly. We saw the Eulerian path algorithm, and phylogeny we will see something called the neighbor joining algorithm, and then when we are looking at suffix trees and suffix arrays, we looked at this Burrows-Wheeler transform, right. All of these are not given as much emphasis in a typical computer science curriculum, right, but looking at it from a data science perspective, some of these data structures like suffix trees and suffix arrays are so versatile that they would have applications beyond genomics and bioinformatics as well in other domain areas where strings needs to be properly indexed so that you can do some large searches efficiently, large number of searches efficiently, and some of these reconstruction algorithms also, like today we will be looking at neighbor joining algorithms that may be of use in other fields as well. Okay. So this diversity of problems and algorithms is one thing that is attractive about bioinformatics and what makes this as a good field to expose data scientists to. Okay. Having said that, let us just jump into today's topic, which is understanding the evolution of a set of sequences. So we know I mean we are still going through the pandemic, COVID-19 pandemic, and the role of genome sequencing cannot be understated here. Within a few days of the widespread announcement of the disease, we were able to find the causative agent of the disease, right. So this is an infectious disease, this was spreading rapidly, and then within a few days, because of the sequencing technologies, researchers were able to identify that this is a particular disease, a respiratory disease that is caused by an underlying virus, and then they were able to sequence it and then say that it is a family of viruses that belongs to the SARS family, right. we will look into a bit more into the SARS family a bit later on. So, so this type of coronavirus family that this virus belongs to was identified as a causative agent, mainly due to the role of sequencing, because they were able to get the sequences from these initial patients and they were able to verify that all of them had this particular virus in them, and this was also verified repeatedly in different parts of the world, right. And then it is not just finding the origins of the disease or the causative agent of the disease, but also able to find how fast the disease is spreading and what is the strains that are happening along the way, right. So what are the mutations in the genome of this virus, what are the mutations happening in the spike protein of the virus, which is one of the proteins that is initially involved in the virus entering the host cells, right, and how does it affect the transmissivity, the transmission rate of the virus, how does it affect the virulence of the virus, how fatal the virus is, how much severe your disease symptoms are. All of that we were able to understand the genomic underpinnings of that, because we were able to sequence these samples and understand how the sequences of the same virus varies as it travels from one region to another region, transmits from one person to another person, right. And here are some of the strains of this virus that are shown, and then this is basically a tree that says initially there was this 19A and then it diverged into 19B lineage and then 20A strains, and then the 20A became 20B, 20C and so on, right. And the common names that you may have seen in the news is like delta, alpha, beta, delta, omicron variants and so on, right. There was a set of sequences that was measured from, let us say here is India, so maybe from some South India you take some sequences, North India you take some sequences, and then each of those sequences you put it through a whole genome sequencing pipeline. So you get all the ACGT letters that make up all the ACGT letters that make up the genome of this SARS-CoV-2 virus. Maybe this is AAT, CGT with this T being changed. This may be AAA, CGG with G being changed. This may be the same as the first virus, right. You have all these different samples from different parts of the region that you are interested in, and then you convert this is sequence 1, sequence 2, sequence 3 and sequence 4, you convert it into a distance based matrix, a 4 cross 4 distance based matrix. Looking at these first few letters, there is no change between S1 and S4, so S1 and S4 will have a distance of 0, S1 and S2 will have a distance of 1, S1 and S3 will have a distance of 1, and this will be 0 of course, S2 and S3 will have a distance of 2 and so on, right. So, this is called a distance matrix. These are the initial sequences. Sometimes these are done over protein sequences, right, by looking only at the spike protein, which is the protein in the virus that is involved in entering the human cells, right. And so, but here for simplicity I am just showing DNA sequences, and then you can look at these distance matrix, and from this distance matrix you want to run through these phylogeny reconstruction algorithms. To get a tree like this, since there are only 4 sequences, maybe these sequences map to these, and this is the tree that you are getting. Okay. If you have 7 sequences, then you may get the larger tree. Okay. So, this is what we are going to be interested in. How do you reconstruct the phylogeny reconstruction algorithms to go from a set of sequences or the distances between all pairs of sequences all the way down to the tree that will help us answer many questions about what the evolution of the virus is, how is it transmitting, what are the origins of the virus, and so on. Okay.